Hi, my name is Jennifer, and this is Jasmine, Mitchell, and Annie. We are all principal investigators on the San Marino High School Mission 3 team, and our experiment is on the effect of microgravity on the antibiotic resistance of P. aeruginosa. So for some background, in 1970, Fred Hayes was infected by P. aeruginosa on the Apollo 13 because of a contaminated water supply. Spaceflight is known to decrease the human immune response because it affects the T cell production and usually decreases the ability for it to be effective.
Pseudomonas aeruginosa is a facultative anaerobe, which means it can survive with or without oxygen. And it has an innate resistance to various antibiotics and can also easily develop new resistance. It's often found in soil and requires very few nutritional requirements to survive and affects those with compromised immune system or exposed tissue. So the objective of our experiment is to determine what the effect of microgravity will be on the antibacterial resistance of this bacteria, which is P. aeruginosa. So the materials we used, we chose to use a type 3 FME, which has 3 compartments. In the main volume, we filled it with a broth medium, which included peptone, chicken extract, sodium chloride, and arabinose, which allows for the bacteria to grow. And in short ampule A, we placed the freeze-dried bacteria, and in short ampule B, we placed L-valine, which is the growth inhibitor.
For our assembly procedure, we put in our first ampule A, into our Teflon tube first, and then we put our second ampule B, which contained our growth inhibitor. We then added the growth medium and sealed the tube and readied it for spaceflight. For our experimental procedure, we began our initiation phase on day A+1, where our astronaut cracked our first ampule A. This released our bacteria into our growth medium and allowed it to grow. On our termination stage, 2 days after, our astronaut cracked open our second ampule, and that released our growth inhibitor to deactivate the bacteria and ready it for return. Onto return from the space flight, we combined samples from our flight and our home and combined them with isoleucine to reactivate growth. And we plated them on agar along with patches of antibiotics. These were allowed to reproduce overnight, and we recorded the zones of inhibition as our data. According to our data, the zones of inhibition for streptomycin and gentamicin were smaller in flight than that of our ground experiment. For the antibiotic ciprofloxacin, our flight zone of inhibition was actually larger than our ground experiment. This suggests that for our bacteria, the antibiotics streptomycin and gentamicin were less effective, and ciprofloxacin was more effective in space.
So, because of this data, we came to the conclusion that Pseudomonas aeruginosa is more resistant to streptomycin and gentamicin after exposure to microgravity, and less resistant to the antibiotic ciprofloxacin. Pseudomonas aeruginosa remained resistant to ampicillin in both the ground and flight tests, which was expected because Pseudomonas aeruginosa commonly displays the trait of resistance to ampicillin. Unfortunately, there were some inaccuracies in our experiment. The first one was a deviation from correct experimental procedures. Basically, what happened is that instead of the bacteria being exposed to growth medium and subsequently the growth arrestor, what happened is that the bacteria was actually in spaceflight was actually simultaneously exposed to both the growth arrestor and the growth inhibitor. So therefore, we postulate that actual little growth occurred aboard the space station, and the bacteria, rather than proliferating in microgravity very much, was more exposed to microgravity. Our second inaccuracy actually occurred from a broken Teflon tube, so our outer tube of our FME actually broke aboard the space station. So what we actually found also is that microgravity aids the growth of Pseudomonas aeruginosa. It actually helps it to divide faster. As you can see in our flight plate, we almost have a complete layer of bacterial growth. And what we found in our home plates that were exposed to gravity is that there were actually there were fewer colonies on the plate. And so the reason for this is that in microgravity, bacteria can actually be suspended in their growth medium. And so each bacteria has access to nutrients and therefore the ability to reproduce. So further steps. Luckily, our experiment is going to relaunch this fall. So we're very lucky, and this is exciting because we get to tweak our experimental procedure a little bit. What we're actually going to do is, instead of measuring a zone of inhibition, we're going to measure what's called a minimum inhibitory concentration, which will allow us to determine the exact concentration in parts per million of an antibiotic that would inhibit the growth of P. aeruginosa, and this would allow us to get even closer to determining dosage of antibiotics. And we plan to use an indicator to compare the growth, and then a spectrophotometer to actually measure the % activity of the bacteria. Finally, we also would like to change our antibiotics to correct redundancy. 2 of our antibiotics actually work in very similar ways by inhibiting protein production. So we hope to change one of those antibiotics so we can actually try to zero in on the part of the cell that is most affected while in microgravity. Finally, we would just like to thank the following people and organizations for all their help. We wouldn't have been able to complete this project without them.
Any questions?
Great job and good luck in the fall. Hopefully things go a little bit better for you. One thing about science is sometimes accidents happen and you got to do it again. My question is more historical rather than on your experiment personally. Fred Hayes got ill on Apollo 13. Jack Swigert and Jim Lovell did not, even though they're in the same water supply. Is there any reason? Do you know of any reason why he's the one that got sick?
You know, I actually don't know. We do know it was a contaminated water supply, but it might be that his immune system was already weakened. As we said before, P. aeruginosa actually tends to affect people who are already sick, so in hospitals a lot. People who are already ill will be affected by it, so that could be a reason.
Maybe
that
he was exposed
Fred
to
Hayes,
something before the flight.
Right.
Okay, thank you.
What could you use this experiment for?
So basically what we wanted to do is determine the dosage of antibiotics when we're in space, because eventually, hopefully, humanity will be colonizing space, and we're already sending people to live in space long term. So we're hoping to figure out how disease treatment will change in microgravity.
Hey, great job. And again, good job in adapting to your disasters that happened or whatever. That's part of real science. I'm curious that you only allowed 2 days for the experiment, or planned for 2 days before you put the inhibitor in there. When you have a much longer observation time to look at that impact, what was your reasoning there?
So what we found actually was looking at growth curves of Pseudomonas aeruginosa is after a few days, it actually begins to die out. So we were hoping to kind of arrest the bacteria at its peak growth and not allow for a lot of cell death.
Okay, thank you.
So since your cells can affect humans, did you have to take any safeguards in handling it, setting up your tubes, or doing your analysis afterwards, as far as your personal safety?
Yeah, we were pretty careful, definitely. Sanitization, and we all wore rubber gloves. Fortunately, none of us got sick. We actually had a mishap when the tube was broken and we took it out of the small plastic bag, and pathogenic bacteria spilled everywhere. So that was a little nerve-wracking, but everyone was fine, so it turned out okay.
How could this help us in the future as human beings?
So this could really help us with being healthy in space, basically. And if someone gets sick in space, this is kind of a pioneering step in figuring out, okay, so how much antibiotics do we give them? How do we change the treatment of people when they become ill in microgravity?
That was a great job, Team 1 from San Marino. And I got to say, your graphics were really superb. Just to acknowledge that. Let's thank Team 1 from San Marino, California.